Hi dear students, a warm good morning to all of you. Welcome to the continuation of chemical bonds and biomolecules, and we will be continuing the sessions which on an online mode since an offline class is not available right now. So let's continue with chemical bonds and biomolecules. As I had previously explained to you, a bond is any type of force which will hold the atoms together. And always atoms try to maintain an octet configuration where the outermost shell will be having or will be having obtain 8 electrons in its outermost shell. And why they try to get an octet nature is mainly because the atoms become more stable when the octet configuration has been maintained. So all the atoms which do not have a 8 electrons in their outermost shell will either try to get it from the neighboring atoms, either by sharing mode, or sometimes they might even transfer the electrons from one atom to the other. And depending upon that, what happens, there is what the atoms may stay together, and this force which keeps the atoms together either by the regrouping of electrons to get a more stable configuration is referred to as a chemical bond. Now, chemical bonds are of three different types. The most first one is the electrovalent or the polar or the ionic bond. I have mentioned all these in the previous classes. I'm just helping you to recollect it. And so the ionic bonds are mainly being formed when two atoms together uh, are involving a bonding, and a electron is being transferred from one atom to the other, so that what the one which gets the electron becomes negatively charged, and the one which gives it will become positive charge. Now in the case of sodium and chlorine is having 7 electrons and sodium is having 1 electron at its outermost shell. So that has been transferred from sodium to chlorine, and sodium becomes positive and chlorine becomes negative. That is an example of a what a electrovalent polar or a ionic bond, where the transfer of electrons are mainly been involved. Then the second type of bonding is the one in which, as we all recollect, the sharing of electrons happen between two different atoms. That is, if there is an atom A and there is an atom B, and if they do not maintain the octet, they will share the electrons between each other. Now, if you go to consider this case, oxygen is having 6 electrons, and in order to get a 8 electron missing, it has to what? It needs 8. So it will take 1 electron from hydrogen and 1 from hydrogen, from the next hydrogen. So a sharing of electrons will happen over there. That is an example of a covalent bond. That is, the electrons of hydrogen as well as oxygen are being shared with each other, and the formation of covalent bonds can occur. Now, the covalent bonds, if one pair of electrons is shared, you form a single bond, and if you have two pair of electrons shared, you get a what? A double bond. And if you have three pair of electrons shared, it is called as a triple bond. So the covalent bonding of different atoms with each other mainly involves the sharing of the electrons, and the primary goal is to achieve the set of 8 valence electrons. Now, a similar bond to that of the covalent bond, as we have already discussed, is a dative bond in which the sharing of electrons do happen, but the electrons are being contributed by only one atom. That is the way in which a dative bond or a coordinate bond is different from that of a normal covalent bond. Now in this case you can look, this is a water molecule, and the octet is already been maintained over here by the water molecule, but when it reacts with HCl, a proton comes and gets coordinately linked towards the water molecule to form the hydronium ion. This, in such cases, the proton is being what? The proton is taking or sharing the electrons of the oxygen here. So, proton is not contributing towards the electron, but it is a water molecule's oxygen which is giving the electron towards to this proton. So this is a case of hydronium ions. So these are the different types of bonds which we had already discussed previously in the offline classes. Now, coming to the biomolecules, we all know that they do have covalent bonds also, but they also have non-covalent bonds. Both covalent bonds as well as non-covalent bonds equally play an important role in the maintenance of the structure of biomolecules. But on the basis of energy, uh, the covalent bonds and the non-covalent bonds are being found to be different. Okay, uh, covalent bonds require large amount of energy to be broken down, whereas the non-covalent bonds they require less amount of energy to break down the bonds. And a covalent bond is a type of chemical bond that forms when two atoms share an electron pair between them. And a non-covalent bond is it is either by completely exchanging the electrons between the atoms or by not exchanging electrons at all. In either two different directions, you can get a non-covalent bond. And similar is the case for electron exchanging also. Either the electron exchanging is being carried out in the case of non-covalent bonds. Here, electrons are being shared in the case of covalent bonds. And I told you, chemical bonds, no, the covalent bonds are being found to be strong in nature. And ionic bonds, now for example, the non-covalent bonds, they are of less energy. And they can include different types of bonds like ionic bonds, hydrogen bonds, van der Waals reactions, hydrophobic interactions. These all will come under the non-covalent bonds. Now let me just help you or link these, what is the significance of these covalent bonds and non-covalent bonds. See, when you go to consider the biomolecules, in certain cases, in different physiological conditions, they have to be sometimes broken down and sometimes they have to be built up also. So coming to covalent bonds, in cases, in physiological conditions, not frequently the covalent bonds are being broken. So those atoms which have to be remained bound together and which should not be broken down easily will use the covalent bonds. Whereas those interactions or those interactions between atoms which especially in the biomolecules, which needs to be broken down at different time points, they will include the non-covalent bonds. Okay, and we are going to learn about the non-covalent bonds. Now, I'll give you an example. If a protein a protein structure is being formed, okay, you have carbon linked or different amino acids which are being bound together. That is a peptide bond is being formed. That is a covalent bond. But there are certain other interactions which happen between for the protein conformations or for the secondary structure formation and all that. That has to be broken, that can be easily broken down at some times or that will include the non-covalent bonds. The same is the case of DNA. Now, if you have the carbon chain or the DNA chain which is being formed by different nucleotides, that will mainly include the covalent bonds. But two different chains of nucleic acids, they are being bound together by certain non-covalent bonds such as the hydrogen bonds. And we know for the process of DNA replication, the double-stranded DNA has to become single-stranded at some time points. So much energy is not needed, should not be needed for the breaking of such bonds. So such bonds which are being involved between the two DNA molecules, that would be a non-covalent bond. So that is the difference between a covalent bond and a non-covalent. The interactions which will involve, which require that these molecules mustn't be broken down easily. In such cases, the atoms will form a covalent bond. And when physiological conditions and when it requires that the interactions should be dispersed or it should be broken down easily, the atoms will form a non-covalent bond. So that's the difference between a covalent bond and a non-covalent bond. And coming to the biomolecules, we have different types of non-covalent bonds, and they mainly include the ionic bonds, hydrogen bonds, van der Waals forces, and the hydrophobic interactions. And do remember, the covalent bonds require larger amount of energy than the non-covalent bonds to be broken down. Now, ionic bonds are charge-based interactions. We will be going to the details of that. And hydrogen bonds are between when hydrogen atom is being shared between oxygen, either from oxygen and nitrogen. We will go into the details. And then we have a van der Waals force interactions where a set of electrons are being under the influence of two different atoms. Okay, it's a weak interaction. And then we also have a hydrophobic interaction, which is just the opposite of the hydrophilic interaction. So let's move on to the different non-covalent bonds in biomolecules in this session as well as in the next session. Now, the first type of non-covalent bond called the ionic bond or the electrostatic bond. If you go to consider here sodium, as I had mentioned earlier, it is having a one electron in the outermost shell. And that when it is transferred to fluorine, fluorine will get an octet. So sodium will also become an octet and fluorine will also become an octet if one electron has been transferred from sodium to fluoride. So in such case, what happens when the electron has been transferred from sodium to fluorine, sodium gets a positive charge and fluorine gets a negative charge. And a bonding will happen. Once it becomes charged, the charged or the differently charged molecules will have an attraction towards each other and they form a bonding. That is an example of a ionic bond or an electrostatic bond, as we had mentioned earlier. So this is also a case of non-covalent bonding. So these are reactions. What happens? The valence electrons are being transferred. One or more is being moved from one atom to the other, and usually the one which loses one or more electrons becomes positively charged and it is called the cation, and the one which gains the electron is considered to be as negatively charged and it is called as an anion. And ionic bonds, they are mainly common in crystals as well as in ionized in water. Now these are some examples where sodium and chlorine sodium electron is being moved towards chlorine. In the case of Mg to Mg2, it has two electrons that has been transferred to oxygen. So this is also a case of an ionic bond. Then you have the calcium chloride where the two electrons are being transferred to two chlorine molecules, and these are examples of some electrostatic or the electrovalent or the ionic bonds which we had mentioned earlier. Now let's talk about these ionic bonds within different biomolecules. Now if you go to take a protein, okay, within a protein, the protein is made up of amino acids, and different amino acids are being linked together and bipeptide bonds, that is all there. But attached to these amino acids towards the side chains, there would be certain molecules which are being found to be present. Now look at here. In this amino acid, a lysine over here, you can see there is a NH3 plus or a amino group which is being found attached to one end at the side chain. And if you go to take another amino acid protein chain, you can see if you have aspartic acid, COOO minus is a negatively chain. So one is positive charge and one is negative charge. A force of attraction will happen between these two amino acid residues which have been found at different parts of the chain. This is what I have shown you. If this is a polypeptide chain, at different regions you have different amino acids, and at their side chains they would have a positive charge and a negative charge. Why they become positive and negative? To maintain the octet rule, they will have a thing, and they will transfer the electrons, and as a result, once they become positive and negative, they will have an attraction with each other. That is an example of an ionic bond which is being found within the protein molecules. Now, in the biomolecules, in other biomolecules, for example, which we have is in the DNA. In the case of DNA, DNA is being found to be negatively charged, and DNA to be organized within the cells, it gets interacted with certain histone molecules, and these histone molecules are being found to be positively charged. So the negatively charged nucleus DNA, is been interacting with the positively charged histones mainly by ionic interactions. And ionic bonds are being formed between them. I hope you got an understanding of the ionic bonds right now. And another example of ionic bond formation within the biomolecule is in the case of enzymes. Now, for example, if you're having an active enzyme and this is the active site of it, and in the active site, probably you see if there is one moiety which has been found to be positively charged, okay, and there would be one moiety which has been found to be negatively charged. The substrate can come and interact with the active site only if the what happens, the substrate and the active site have opposite charges, or the moieties have been having, only then the correct configuration or the correct binding of the substrate will happen at the active site. And now if the pH has been changed or something like that, in such cases what happens, the charge variation will happen and the unbinding or the proper binding of the substrate to the active site will not happen. So ionic bonds play an important role in different biomolecules, let it be proteins, nucleic acids and all that. The binding of these, either the substrate or the proteins to nucleic acids as such, all that is all being carried out by the ionic bonds. And do keep in mind, the ionic bonds are non-covalent bonds and they do not require much energy to be broken down. And that is the reason why at some stages the substrate is able to bind with the enzyme, and some stages they are moving apart. And now in the case of DNA, we know when each time a cell is dividing, it requires a DNA to be what? Unbound and again bound. So the reaction of winding and unbounding has to be carried out in a frequent manner. And for that, a non-covalent bond, such as an ionic bond, which requires less amount of energy, is being utilized. I hope you got an understanding of the ionic bonds. We will be moving on to the next bonds in the next sessions. Thank you for now.